Hello, everyone. Welcome and thank you for joining today's webinar, Particle Size Analysis of Polydisperse Liposome Formulations with Multispectral NTA, Nanoparticle Tracking Analysis. My name is Julie Chen Nguyen. I'll be facilitating today. It's my great pleasure to introduce you our speaker, Dr. Pushpendra Singh. Dr. Singh is a biomedical scientist with more than 15 years of research experience. He received his PhD in biochem and biophysics at Johns Hopkins University. He is currently working on the development of liposomal adjuvant towards development of vaccine against HIV and other infectious diseases. We'll get to learn more about that study today. Okay, Dr. Singh, I'm going to go ahead and pass the ball to you. Would you please share your screen?
Sure. Thanks, Julie, for the kind invite and the kind introduction. Today, as Julie mentioned, I'll be talking about this new novel biophysical technique of multispectral nanoparticle tracking analysis, how we applied it to our polydisperse liposome formulations that we made in the lab. Okay, so I have divided the whole talk in four subsections: liposome and their applications in the medicine, the liposome manufacturing process, which will entail us that how these particle size heterogeneities are originated in the liposome samples. They are polydispersed, they could be monodispersed, polydispersed. By polydispersed, what I mean is the heterogeneity in the particle size distribution, and monodispersed, I mean that they are quite sharply distributed in their range of size. Then I'll talk about why do we at all care about this particle size distribution, especially in context of the uh, liposome-based vaccines, since we make vaccines in the lab and we are running several clinical trials here at MHRP at Walter Reed Army Institute of Research. I'll take you through to the challenges that we face through the current particle sizing techniques and how multispectral nanoparticle tracking analysis has been useful, fruitful for us to give us adequate particle size distribution for polydisperse liposome formulations. All right, so to the first part, here I show you this table in which marketed liposome-based therapeutic products have been detailed. You may not see all the products here, and there is a lot of text on it. What I want to convey here that many such products have been already in the market, FDA approved, and so many more are coming. They are undergoing phase 2 and phase 3 clinical trial. What you see here in the right hand side is a typical liposome. They could be conventional, as shown in the left hand corner of the slide, or they could be surface modified by pegylation, which is pegylated liposomes. These liposomes can be targeted to a specific tissue and a region in the body by using some antibodies or markers against by functionalizing them further. A field of theranostic liposomes is coming up. So what I mean by theranostic, therapeutic and diagnostic. So it's a merger of therapeutics and diagnostic, which has been named as theranostic. So liposomes are being used for both therapy and as well as diagnosis of certain diseases and disorder states. Liposomes are heavily being used in pharmaceutical industry as drug delivery system as well as vaccines, and I'll be talking on the vaccine part more later in my talk. So liposomes are a specialized delivery vehicles. They mimic more on the biological membranes and they are recognized more in the cells. They have the lipids and the proteins inside, depending on what kind of liposome you are talking about. But generally, they are made of lipids. You can put proteins also. Generally, liposomes take some drug and they actually protect it from degradation by the action of hydrolytic enzymes which are there in our body, and so they keep them in circulation for a long time so that they can be more effective, and that one way they actually enhance their efficacy in the body. Further, they possess the unique ability of encapsulating hydrophilic as well as hydrophobic drugs. They can solubilize hydrophobic drugs. There are examples in the literature where liposomes have been used to solubilize drugs about 50 to 60 folds in the literature for the lipophilic drugs, which are not anyway soluble and they aggregate when we inject in body. By doing that, they have actually increased their efficacy and they are being much more effective at lower concentration, lower dosages. So what I want you to take away from there that liposomes are very useful in modulating the pharmacokinetics and biodistribution of the drugs, and that way they minimize the side effects in our body. Pharmacokinetics is something is like how long a drug sustain, and a biodistribution is where in the body it gets distributed, and these are two important parameters for any drug and vaccine to be effective in our body. Liposome, as you see from this diagram, is a spherical ball of lipids where lipids which has two faces, one is hydrophilic, other one is hydrophobic tail. They actually assemble non-covalently, they self-assemble non-covalently form a bilayer sheet here, and then they actually encapsulate some of the aqueous solution, and in exterior also of this hydrophilic region face the aqueous solution in the for liposomes. So this is the typical architecture of the liposome shown in these diagrams. Liposome manufacturing process include three important steps. One is drying of the lipids, other one lipid hydration, and the critical third is the most critical point is the downsizing of the lipid vesicles. So I'll go through them one by one. So lipid stocks are generally made in organic solvent. Most often it is a chloroform, but you can use methanol and ethanol also in some cases. Once you actually make lipid stock, then you mix them in desired concentration that you want to achieve. And then these lipids, which are actually mixed together, are dried up. And the way to dry up is to evaporate this organic solvent. So in the lab, we use a rotary evaporator. In this, we create vacuum, and by creating the vacuum in the vessel, then the organic solvent, which is already a low boiling solvent, start boiling further at room temperature, at lower temperature, and then these vapors are condensed further and collected as a waste in here. What you are left with is the dried lipid film. There could be other methods also of evaporating organic solvent, like you can flush inert gases like argon or nitrogen to evaporate the organic solvent, but they are done more at the small scale, while rotary evap can be tried for the large scale preparations. Anyhow, you get with this dried lipid film that you can swell in the aqueous solution. So aqueous solution could be a buffer of a defined pH, and depending on what kind of lipids and what kind of formulation you are planning to create, you can choose your buffer of choice. So if you are having pH switching liposomes that actually act upon changing the pH, you can have different pH of your buffer. Generally, the liposomes which we prepare in the lab have pH close to the neutral pH, like pH 7. Then, when this dried lipid film is swelled in aqueous solution, you can start agitating it. So this agitation can be done either by vortexing it through handheld vortexer, or you can use more defined way of agitating it, where you can run that on a constant speed, so that you can more defined multilamellar vesicles, in other words MLVs. And these MLVs are large size multilamellar vesicles. As you see from the cross section of the MLV, this is a cross section of MLV showing you multiple bilayers, a vesicle inside vesicle. So that kind of arrangement is generated upon agitation of the lipids which are swelled in the aqueous buffer. Then the critical steps is the downsizing of these MLVs to generate large unilamellar vesicles or small unilamellar vesicles. That can be done by using some of the downsizing technique, which I'll be talking in the next slide. But once you generate these large unilamellar vesicles or small unilamellar vesicles, you can sterile filter them if you want to use for medicinal purposes, and you can actually keep them in a sealed vial or in a sterile vessel. So there are three main downsizing methods for liposomes: sonication, extrusion, microfluidization. The thing which is common in all these downsizing methods is that they impart energy to break down large multilamellar vesicles, MLVs, into small unilamellar vesicles. And depending on the energy, how much, and depending on the arrangement you have for these methods, you can generate liposomes of desired size. Like, for example, in sonication, you apply ultrasonic waves either through the probe sonicator or through bath sonicator that result in the breaking of these MLVs to generate small unilamellar vesicles of generally of 50 to 60 nanometer size. But again, it depends on how much energy you are imparting to actually attain one kind of liposomes, one kind of size of liposomes. Extrusion involves defined size filters of polycarbonate, which can be used like for 100 nanometer or 200 or 400 nanometer size of vesicles. So what you do here, you take colloidal solution of MLVs in one syringe and pass through the polycarbonate filter multiple times to get homogeneous size distribution of the liposomes of defined size. Microfluidization is the technique we use in our lab very routinely, and there you apply microfluidization high pressure to break down these large vesicles into small. So microfluidizer is something, it's a lab scale microfluidizer I'm showing you here. So you apply this colloidal solution in one syringe as an input, and then you get the desired product here. So in this scheme is the inlet reservoir which contains colloidal particles, and then at high pressure they are broken down into small homogeneous solution, which is a true solution here. So what happens, this is a colored solution I'm showing you here, but for liposomes, when there is no dye in there, these are multilamellar MLVs in the aqueous solution. So when you apply high pressure in there, these MLVs are broken down into small vesicles, small size vesicles, and this is a milky solution which becomes transparent. So, and this is because the size has been gone down to less than 100 nanometer, which cannot be perceived by our naked eyes. So typically, the distribution of particles in these multilamellar vesicles and small size liposomes differs. As you see here, this is the light microscopy showing you the small size as well as large size vesicles, and this is the frequency distribution of them, showing you that there's a lot of heterogeneity in this solution, this sample, where you have vesicles ranging from hundreds of nanometer to tens of micron. While in case of this transparent solution, which contains only small size liposomes, you hardly see any small size liposome by optical microscopy. While by techniques like dynamic light scattering, you can see easily a very sharp, tight distribution of the liposomes in the range of 100 nanometer. So I like to now switch a gear about the liposomes application in the vaccine. So liposome has been used as adjuvant in the vaccine. Adjuvant is something that enhance and direct the immune response to a specific antigen in the vaccine, and that's the way it actually boosts the immune system to produce more antibodies. Our lab has created army liposome formulations named as ELF, and these ELF types of liposome are many in the lab now. There are many subtypes, and that depends on the concentration and the combination of immunostimulatory molecules inside them. So, for example, monophospholipid A, most commonly called as lipid A, and saponin QS21, and aluminum salt, aluminum hydrogel is the most common that we use. So depending on their concentration and combination, these ELF type liposome can be further defined. So historically, the vaccine design have been from the live pathogen that you create this attenuated pathogen or weakened pathogen by killing it using heat or some chemical process, just to kill the replication efficiency, replication potential of this pathogen so that it can stay dead, but keeping all the immunogen around it. And that has been historically the vaccine. But we realize over the period of time that these vaccines, though they are not able to replicate, but they are still toxic and they can be pretty bad in some cases. So there has been always a drive to produce safer and safer vaccine. In that process, we produce this so-called third or fourth generation vaccine, which contains this recombinant immunogen to the purity, so they do not have any bad impact on the immune system. So they are very, very safe. But at the same time, in the process of increasing the safety profile, we learned that the immunogenicity of these immunogen is quite weak when we take recombinant immunogen. So to deal with this problem, we researchers came up with this solution that when to make a balance of making this vaccine immunogenic so that it can protect us without compromising safety. There is additional molecules which were discovered and that they can boost the immune system. And these are the one which we name as adjuvant. And adjuvant is the molecule, as I said previously, that enhance the immune response, that boosts the immune system to produce more antibodies and cytokines against a specific antigen. All right. So these adjuvants are being used increasingly, though they were discovered. Their first use was in 1920s, and after that there was a break up to 1980s. There were not much of them, much going on here. So these vaccines were not having these adjuvants, but right after 1980s, every other vaccine has an adjuvant. And this happened because there were more immunostimulatory molecules. There were a lot of research done on that, and there were specific immunostimulatory molecules were discovered, like which are agonist against TLR, this toll-like receptors, and then there were like other that can boost the immune system by other mechanisms. And now we know at 2010 there are more than 500 vaccines which contains adjuvants. There's something remarkable happened in 2017, late 2017, where this vaccine was licensed by FDA to GlaxoSmithKline company. So this is trade name is Shingrix. This is the most successful vaccine against shingles disease, and lot of credit is given to the liposomal adjuvant in it, which is called ASO1B, adjuvant system O1B. So it's very similar to the ALF vaccine, Army liposome formulation vaccine, of Army liposome formulation adjuvant that we have in several vaccines. ALF has been actually used in many vaccines. So we have used in several experimental vaccines against malaria, ricin, Ebola, anthrax, HIV, dengue, and cancer. And by its success, by the result of this ALF in several these anomalies, we found out we went on to use it for 14 phase 1 and 2 phase 2 clinical trial, and that's showing a very promising results there. So now it's being used for malaria, HIV, cancer, and meningococcus. So the core composition of ALF has been these four lipids, two phospholipids, DMPC and DMPG. DMPC is a 14 carbon saturated deuterionic phospholipid, while DMPG is 14 carbon saturated anionic phospholipid. And cholesterol is an important component of these ALF formulations. And the most important is the monophospholipid A, which is an endotoxin isolated from the LPS of bacteria, lipopolysaccharide of bacteria. And this is the immunostimulatory molecule in ALF formulations. Now, we have these four types of ALF. They differ in chemical composition. ALF contains the MPLA. ALF A contains aluminum salt in addition to MPLA. QS21 is present in addition to MPLA in ALFQ, and the ALFQA contains QS21, aluminum salt, and MPLA as an immunostimulatory molecule in it. And they differ chemically, and not only that, their biophysical size, biophysical properties are also different. Particularly particle size distribution we have found varies quite a lot among them. And that's where we had the need of a particle sizing technique that can measure the particle size distribution accurately so that we can actually produce stable vaccine formulations. And this is one of the FDA requirement we had. So in order to do that, we look for many techniques, and then we found out finally that there is a new multispectral nanoparticle tracking analysis, which was by Manta. Now it's part of HORIBA. So we established this standalone method for particle size characterization, and we found it very useful for our polydisperse liposomal formulations, which are used in vaccines. So why care about the particle size? So as I said, that the vaccine in application of this alpha vaccine has been quite promising, and we wanted to define the particle size distribution. Decades of research has shown when you have identical chemical composition for a vaccine formulation and just vary the size, the depending on the size of the size of the particles in the formulation, their distribution and kinetics in the body are different. Small particles tend to go to the bloodstream rather fast, and then while large particles stay at the injection site for longer time. Not only that, their processivity is also different. Small particles tend to picked up by these special antigen presenting cells like dendritic cells, and then they are processed differently, while large particles are taken up by these macrophages and monocytes, which process them differently. And hence the size of the particles even in the identical chemical composition can lead to a different immunological outcome, and that's what we know. In case of ELF, we have some data like when we have this formulation ELF which contains identical chemical composition for ELF, and we create these two formulations which just differ in the particle size distribution. ELF wet has only small size liposomes while ELF Llyo has small as well as large liposomes. This graph is just showing you the large particles because here dynamic light scattering graph do not actually take care of the small size particles when they are mixed with the large particles, and because they are polydisperse in nature, and that is indicated by the high polydispersity index PDI for this formulation. So these results are not quite reliable. So in order to look at the size distribution of this formulation, we used EM. So electron microscope shows a monodisperse homogeneous distribution of these particles in ELF wet, while they are quite large to small size in case of ELF Llyo, suggesting that they are polydisperse in nature. So when we use identical amount of ELF wet and ELF Llyo with two different antigens, we end up having two different results. So here you see that the endpoint antibody titer against this tetanus toxoid is very similar when we use ELF wet and ELF Llyo liposomes. But in case of GP140 antibodies, which is a HIV envelope protein, ELF wet seems to be doing much better job than ELF Llyo. There is some unpublished data which I'm not sharing here, where we have seen that ELF Llyo works better than ELF wet in certain cases. So what to take out from this slide is that one size does not fit all. So depending on the antigen, the size distribution of the ELF formulation matters. So you need to actually customize each adjuvant for each specific antigen probably. And for that, we need to know the exact particle size distribution of the formulation. And so we cannot achieve with the current particle sizing methods. So what are the challenges we faced? I'll like to go through that so that we can appreciate that what we are doing with multispectral nanoparticle tracking analysis. So these are three popular particle sizing techniques used in the field: dynamic light scattering, laser diffraction, nanoparticle tracking analysis. So in dynamic light scattering, you have the light scattered intensity, which is recorded. So this is the light scatter intensity from the particles which are undergoing binary motion. So large particles tend to give you the broader distribution, while small particles give you much sharper distribution. And then these fluctuations are mathematically autocorrelated to deduce the size of the average size population of the average particle size. So DLS works in the range of 30 to 10,000 nanometer, pretty good range. But as I said, it does not work well on the polydisperse sample when there is a presence of large size liposomes, large size particles. They tend to mask the abundance of small size particles, and hence this is not the choice technique for our formulation. Laser diffraction, on the other hand, collects scattering intensity at different angle, and from that information, depending on the size of the particle, their angle of scattering is different. And from this diffraction pattern, you can deduce the average particle size. It works pretty large range from 10 nanometer to 5 millimeter. Also, it has advantage of working on the not only the aqueous solution but on the solid particles too. And it does not work well for our polydisperse sample for the reason that large particles tend to mask the presence of small particles. NTA, nanoparticle tracking analysis, the way NTA works, it's very analogous to microscope. So you are looking at the particles which are undergoing Brownian motion and you are recording their movement, and then later on you are tracking them one by one. And from there, the particles which are moving fast are likely to be slow, and the which are moving slow are going to be large in size, and that's based on the assumption that they are all undergoing Brownian motion. And this works very well in determining the particle size of individual particles in the formulation, but it works only for a small range from 30 to 1000 nanometer. And but it works on the polydisperse sample, and we have seen for our liposome formulation, and that's where I spent some time to talk about our liposome formulation, that they have significant number of particles after 1 micron. So you are ignoring a lot of particles beyond micron size. So then we come up with this multispectral nanoparticle tracking. Also, a trade name is ViewSizer 3000, and this is the machine showing you, and this is the snapshot of the video. So the way it works is very analogous to NTA, except it has three lasers simultaneously looking at the particles so that you do not miss any information. You collect information from small to large size particles. In addition to that, it has a multispectral detection system, so you can detect all the colors which are being diffracted from the particles. Then you use an algorithm to track the particle movement in the recorded video, and from there you get this trajectory of the particles. And from this trajectory, you can calculate mean square displacement, diffusion, and the size of the particle. So that's how multispectral nanoparticle tracking analysis works. So this is a snapshot of 70 nanometer size polystyrene latex beads showing you the distribution of the particles in the optical setup of the ViewSizer. And you see that they are pretty homogeneous, which is what you expect when you are having one size beads. But we also created the mix of the beads, and that mix we tried with different techniques. So I'll be talking about the comparative analysis using DLS, laser diffraction, and nanoparticle tracking analysis. So as you see in the DLS, four beads of 70, 200, 600, and 1300 nanometer size were identified accurately when they were used alone. You get an average size, but you do not see the individuality of these beads, which is something is expected from DLS. For laser diffraction, again, you see the individual particle size correctly when you use only one bead, but when you mix them, you get an average size more similar to DLS, suggesting that they do not, give you the size distribution for these size ranges when they are close together. In case of NTA, you do see the particles individually in alone as well as in the mix. The only thing is that this 1300 nanometer size range, which is beyond 1000 nanometer, is identified incorrectly. Actually, incorrectly, you have the shoulder coming up, and then this size is actually identified as 1 micron, which we know is not correct. Then we use this multispectral nanoparticle tracking analysis, which was named as multispectral advanced nanoparticle tracking analysis, sorry, MANTA, and that's what we have maintained that in our paper that we published recently. So here you see that not only the individual size were identified, but also these peaks were actually identified in the mix also, suggesting that MANTA is much more superior technique as compared to them when it comes to the individual particle size distribution, and it goes beyond a micron size particle. So we use to sample liposomes in our application, LVET and Llyo. LVET is analogous to alphabet that I talked about initial in the beginning of the talk, and Llyo is more analogous to Llyo. So LVET is prepared by microfluidizing the MLVs to generate uniform monodisperse small size liposomes, while Llyo is prepared from LVET by lyophilizing it. In the lyophilization, you are evaporating any aqueous solution out of it, and then you are left with lipid cake, which is again reassembled in the aqueous solution, and then you generate liposomes from nano to micron size because you are left with the powder after lyophilization, and by adding water is more analogous to the swelling process, and that's where you get more nano to micron size particles in there. So it's quite heterogeneous and it's polydisperse. So to make measurement very accurately, we made the measurement, blank measurement on the diluent, which is PBS in this case. And as you see here, that there are very few particles present, and that's what you want for your application because you do not want too many particles in there because they will spoil your measurement. In fact, we have seen that we have cutoff of 30 nanometer size, so any particle less than that is excluded as impurity. It could be some bubble which we see in there. Same thresholding was applied to later on. So before actually making the measurement, we determined the linear range of sensitivity for these two different formulations. For LVAT, this sensitivity linear range was quite broad, while for Llyo, this was quite small. So we worked with this window in Llyo and as well as in LVAT so that we get consistent results. Because if you want to have very reproducible results, you need to maintain the range of sensitivity for your optical system, and it depends on your sample. These are the snapshots showing you for the LVAT and Llyo. LVAT, you see there is a more homogeneous size distribution as reflected from the intensity, scattered intensity for LVAT. Llyo has small as well as large particles in it. So LVAT is monodisperse, homogeneous in other words, while Llyo is quite polydisperse. Okay, this is the snapshot showing for LVAT, and the way we processed it, we did not take anything less than 30 nanometer into account for the particle size distribution for LVAT, and you see that the distribution is quite sharp. In case of Llyo, here again we maintain the same threshold of 30 nanometer, and then you see that there is a lot more particle beyond 1 micron, and they are declining in number, and there are hardly any particle at 2 microns, suggesting that our choice of technique is pretty good and covering majority of all the particles. So these are 4 different techniques applied on the same samples. LVAT is monodisperse and there is no problem in detecting the size of this sample by any of the technique, while Llyo, which is polydisperse in nature, you say DLS give very aberrant result, while laser diffraction only identified large particles, which is also due to some complex interaction with the large and small. So let's say there was one particle that masks all the other particles in there, which are smaller in size. While NTA gave again up to 1 micron. This is reliable, but this also gave very discrete sizes, which can be talked about. In case of Manta, we see a very continuous size distribution of the particles, which is something stochastically expected for a non-covalent assembly like liposomes, and this goes down to 2 micron, and you see very few particles beyond 2 micron, suggesting that we are covering almost maximum size range of liposomes in Llyo in polydisperse samples. So Manta is a choice of technique for measuring liposome formulations like we prepared in the lab, and this is quite good. So just to summarize this, multispectral nanoparticle tracking analysis can measure particle size very well between 50 to 2000 nanometer in polydisperse liposomes, and it works well with the lyophilized liposome formulation that we have, and we determine that more than 90% particles are in the range of 500 to 1500 nanometer size range for polydisperse liposomes LVAT that we have used. One thing I like to mention here that we have used only nanoparticle tracking analysis application of multispectral nanoparticle tracking analysis, but it has a separate application which is sinking mode. And sinking mode is based on the Stokes law principle. So there you can use this application for the formulation which contain really heavy particles which are actually settling down in the time of experiment, like gold particles which are quite heavy. And there the range can be further enhanced, and it is promised to actually work up to 15 micron size range, which is quite huge. if the particle shows the sinking in the time of the experiment. Also, if your particles contains very good high refractive index, then NTA can give you much better results also. But for polydisperse liposome, we saw that this works well up to 2 micron, and we cover most of the particle size, and that's where we are confident that this is an application of choice for our vaccine formulation that we will generate based on these liposomes. All right. So with this, I have come to an important slide where I need to acknowledge the people involved. I like to thank Gary Matias and Jolton Beck for their constant encouragement and support and the independence they gave me to execute my plans and ideas while I was working on this technique. I like to thank people from HORIBA, mostly Jeff, who has been instrumental in carrying out all these experiments, and we worked very closely to tackle all the issues that we got in software or hardware, as can happen with any technique. Kuba for explaining beautifully the principles behind it and have been the force behind this technique to actually go forward. So I'm really grateful to him. I like to thank Bill and Sion from Anatome, who have been great colleague to help me out whenever I ran into any issue, any problem with the technique. And Bill helped me in making some measurement on the laser diffraction. So it's a great thing to work with them. I like to thank leadership of Rare, Nelson Michael, Robert Gramzinski, LTCP Julie Egg for maintaining the facility here. I like to thank HJF leadership, Sandhya Vasan and Merlin Rowe for maintaining facilities and supporting us constantly. I like to thank Henry M. Jackson Foundation for supporting me financially for this work and for me. And with this, I'm happy to announce that we just published this paper, and there would be more detail that can be found out from the paper. Very happy to take any question. Thank you.
Thank you. Thank you, Pushpendra, for the excellent webinar. Um, I don't have any questions at the moment, but I did have a question that came in during registration. I think this person wanted to know what is the feasibility of particle size measurement in non-aqueous medium?
That's a great question, and I think the technique of choice so far has been laser diffraction in there, where laser diffraction you can use it as I mentioned in my presentation. You can do it for aqueous as well as powder form of the sample. So laser diffraction at present is the technique of choice right now. I do not see MANTA is always multispectral nanoparticle tracking analysis always require a Brownian motion because and that can happen only when you have a molecule in aqueous solution and then they don't interact with the material of the cuvette and among themselves. But for powder form, I would encourage to use laser diffraction from HORIBA again. That's an excellent tool to be utilized.
Thank you. Yes, so the laser diffraction instrument that Dr. Singh is talking about is the LA960, and we would be more than happy to take a look at your sample, just like Dr. Singh did with the ViewSizer 3000. Don't hesitate to reach out to us at labinfo@horiba.com. And I think I also wanted to add that I think previously you mentioned about the syncing mode. Essentially, that's the mode where the software uses Stokes' law to look at sedimentation as opposed to the typical NTA and nanoparticle tracking analysis. So that's an additional software capability that the ViewSizer offers. For now, thank you so much, Dr. Singh. Have a great day, and we'll see you at one or more of our future webinars.
Thank you so much, Julie. Once again, I thank you for taking care of it very nicely.
Absolutely. My pleasure.
Yeah.
Thank you so much.
Bye.
Thank you.